I just published a paper about my PhD research. It's called Complex Developmental and Transcriptional Dynamics Underlie Pollinator-Driven Evolutionary Transitions in Nectar Spur Morphology in Aquilegia, parentheses columbine. I wanted to call it Columbine Flowers Are the Coolest. I was not allowed. But that's basically what the title is saying when you break it down. So columbine flowers, aka the genus Aquilegia, make crazy petals. Look at them! They're nothing like your typical paper-thin, flat rose petal. Columbines make 3D petals. The 3D structure is called a spur. It secretes nectar as a reward for pollinators. For this paper, I studied two different nectar spur morphologies, or nectar spur shapes, that correspond to which animal pollinates a given Aquilegia species. Species with purple curvy spurs are pollinated by bees, while species with red straight spurs are pollinated by hummingbirds. The first ever columbines to evolve were bee-pollinated with curvy spurs. But then there were two completely separate evolutions of hummingbird pollination. That means red flowers, straight spurs, changes in nectar chemistry. These all evolved twice independently in the genus Aquilegia. What? I want to study that. How'd that happen? So to find out, first I looked at spur development in curvy and straight spurs. What's happening as a spur grows to get these different shapes? Here's a curvy spur. For the rest of the video, I'll be referring to this surface as distal. This is the farthest from the center of the flower, and the opposite surface as proximal. It's closest to the center of the flower. In order for a spur to be curvy, the distal surface has to grow more than the proximal surface so the spur can bend around like that. And in straight spurs, growth has to be relatively even between surfaces so the spur remains straight. Well, what do those growth patterns look like at the cellular level? To answer that, I counted and measured the cells along the entire proximal and distal surface of curved and straight spurs. Sounds simple, right? I had to collect a ton of petals. Shout out to the Mass Audubon Wellfleet Bay Wildlife Sanctuary for letting me sample your flowers. The chemical preservatives I collect the petals in bleaches the petals. So next I had to stain them so their cells would be visible again under the microscope and squish them so they'd be nice and flat. For each petal, I took dozens of photographs on the microscope to capture the entire outline of the spur, then counted and measured every single cell along the spur perimeter using a computer program. It wound up being tens of thousands of cell measurements. In the paper, I present data from multiple developmental stages and species, but the main take-home is that curvy spurs are curvy because cell division lasts longer in the distal surface than the proximal surface, so mature spurs wind up with more distal cells than proximal cells. So we thought, okay, well, maybe straight spurs evolved by adding more cells to their proximal surface. But no, we were shocked to find that in both of the independent evolutions of hummingbird pollination, straight spurs still have way more distal cells than proximal cells, just like their curvy ancestors. Instead, their proximal surface cells are longer than their distal surface cells to compensate, which evens out the overall lengths of the surfaces and generates a straight spur. So this was already turning out to be a way more complicated and interesting story than I was expecting. Once I understood the developmental dynamics of spur shape, I wanted to see how they play out transcriptionally, which is just a fancy way of saying, what's going on with the RNA? You might already be familiar with DNA, the molecule that encodes our genes. All cells in a columbine plant have the same DNA, the same complete set of columbine genes, but not every cell in the plant is expressing the same genes. A cell in the root, for example, is going to express very different genes from a cell in the leaf. They have different jobs, they need different genes for those jobs. Expressed genes are transcribed from DNA into RNA. So if we sequence a nectar spur's RNA, we'll know what genes are being expressed and at what level they're being expressed in the nectar spur. And by comparing gene expression across nectar spurs with different shapes, we can gain insight into the genetic basis of the developmental phenomena we characterized in the first part of the paper. For this RNA sequencing project, I sampled two closely related species, straight-spurred Aquilegia canadensis and curvy-spurred Aquilegia brevistyla. I sampled them at 4 developmental stages, which were based on % of mature spur length. Here are the petals at a slightly larger scale. I removed their blades because I was only interested in gene expression in their spurs, and then separated the proximal and distal compartments because I wanted to see whether gene expression is different between them. Okay, this is a super pretty picture, but what does that actually look like in real life? First, you need to spray down literally everything you'll be using to dissect your spurs with a chemical that will prevent RNA from getting degraded. RNA is super delicate, so we have to do everything we can to make sure it doesn't break down during our dissection. Then we set up an elaborate dissecting surface on ice, again to preserve the RNA. Shout out to my lab mate Steph for developing this setup. Then, one at a time, I dissect each spur and collect the proximal and distal compartments into separate tubes. The tubes are suspended in liquid nitrogen, which is so cold it immediately freezes the spur tissue on contact and preserves the RNA. Columbine flowers have 5 petals, so I would pool all 5 spur halves from a single flower into a tube. And I collected 5 replicates of each stage and species. So there were 80 samples total. 2 species, 4 stages, 2 tissue types, 5 reps. And then we extracted and sequenced their RNA. With a sampling scheme as complex as this, there are seemingly infinite ways to analyze and explore the data. There are 7 figures and a dozen supplemental tables in my paper detailing the results of all these analyses. You're welcome to check them out, but we don't have time to dig into that many heat maps and gene lists today. I just want to share some key takeaways that are relevant to our main story. Namely, there was loud and clear developmental signal in this RNA sequencing data that reflects our findings from the first part of this paper and helps us understand the molecular processes underlying them. Overall, we found that the proximal and distal compartments of the spur are experiencing very different gene expression environments, with several thousand genes showing different expression levels between them. That was very surprising given these tissue types are coming from the same organ. They're all petal spurs. The big result from the first part of our paper that explored spur development was that cell elongation in the proximal surface was key to the evolution of straight spurs. And our RNA sequencing data supported this finding, with lots of transcriptional evidence for cell elongation and cell wall synthesis in canadensis proximal tissue. We also found repeated indications that a hormone called auxin could be involved. Auxin is a known promoter of cell elongation across the plant kingdom, and lots of genes contribute to its synthesis and regulation. Finally, we combined the data for this paper with data from my first author paper to identify several genes we think could play a role in spur shape. We're super excited about a gene called IBL1, which we think might control the curviness of the spur. We can't wait to test this hypothesis and explore the function of IBL1 further. On the whole, this research painted a rich and dynamic story about spur shape in Aquilegia. We already knew they had crazy petals, but figuring out the details of how they developed and evolved revealed a more complex story than we could have imagined when we first set off on this project. Maybe an even better title would be Cool Columbines Are Even Cooler Than We Could Have Thought. So that's the science, and now I need to take a minute to talk about the people behind it. This paper was part of my PhD research, and the other 2 authors on it are my advisor, Elena Kramer, and my mentor and close collaborator, Angie Ballerini. They guided me through this beast of a project and helped me tame a story out of 2 very large and unwieldy data sets. I couldn't have done my PhD without them, and I love and appreciate them very much. And I am so proud to publish my paper in the American Journal of Botany, which is the journal of the Botanical Society of America. BSA has been my scientific society since I was a baby botanist back in 2013, and it means so much to publish my dissertation work with them. For more botanical adventures, please subscribe. A link to the paper is in the description, and I'd love to answer any questions you have about it in the comments. Finally, thank you to Svaha for sending me this sciencey dress to celebrate my paper. If you want your own sciencey apparel, check out their link below. See you next time!